So sorry for the delay. Today I wanted to talk to you guys about this project, which I call the Mada-Lichen Project, where we're barcoding we're doing DNA barcoding, which I'll get into in a little bit here, of specimens that we collected in Madagascar. And this was back in 2019 when I collected these. This project should have been done a long time ago, but COVID really slowed things down. So I will proceed here. So just a little background, Madagascar is off the east coast of Africa, and it is one of the world's most endemic regions. 90% of the wildlife is found nowhere else on Earth. And so this goes for their animals, plants, and probably their lichens. And I went to two different places when I was invited out there. One place is called Mbui-Tenteli. It's about a 2-hour drive from the capital, Antananarivo, or as they call it in Madagascar, Tana. And it's in the central highlands, and it has montane rainforests. And it's kind of a fragmented preserve, and it's really threatened by wildfires and slash and burn agriculture and harvesting of trees for charcoal production. So we were out there with a big group and people from all over the western part of the Indian Ocean. So there was people from Madagascar, Reunion, Mauritius, the Comoros. These are all professionals that are like conservation experts, park rangers. They all came for a training session, and they get trained on doing studying birds, mammals, plants, and then lichens was one of the training sessions that I was leading. And so I was taking around a group of people in these montane forests. And it was just, I've never collected in a place like this before, so it was all new for me. And it was all new for them because they had never studied lichens before. And there was a lot of terrestrial leeches here too, which was really unpleasant to deal with, but I survived. They don't carry any diseases like mosquitoes can. And I just have a few pictures of some of the lichens that we saw. I have plenty more. I'd be happy to share them with anybody if you shoot me an email. I have my email link at the end here. And actually, this one on the left is on the global website. It made it online. And the second place that we went to is called Ankarana. And this was like a 2-day drive on very rough roads. And this is a different type of forest. It's called a lowland humid forest. And again, we had a group, a different group. These were students in Madagascar, and they were coming kind of for the same type of training. They were rotating around, studying different organisms, and then lichens was one of those. And they helped me collect a bunch of them, and we had a really good time. Ankarana is a national park that has these amazing rock formations that's all volcanic, and then you have a lot of lichens growing on these rocks, and then also some that are on the vegetation there. And during these 2 trips, we collected 500 specimens, and I carried them back in my luggage, which was, they almost got confiscated by the USDA in Atlanta. And they actually did get confiscated, and I, through lots of phone calls, I got them to release them and give them to me. It was very scary. all that hard work would have been gone. But fortunately, I got them back to the museum. And but the thing is in Madagascar, the lichen diversity is not very well known. There was a checklist made about, I think, around 5 to 10 years ago. It has 500 species of lichens on it. But to put that in perspective, Madagascar is about the same size as California. And California lichen checklist has about 1,500 species on it. So like 3 times as many as the Madagascar checklist. So I think Madagascar is very underestimated from the checklist. And it's just that there hasn't been a lot of people collecting there. And it's not the easiest place to get around in. You got to know people. The permit process is challenging. Hey, Luis. Sorry, I saw somebody who's been a lichenologist friend. Yeah, going to a place in Madagascar and collecting is you're bound to find new species and undescribed things. So what I'm doing now with these 500 specimens that are collected is it's called metagenomic DNA barcoding. And basically we take these 500 lichens. So in the species box there, instead of all these fish and crabs and insects and plants, just replace those all with lichens. And then we get their DNA out and we sequence it. And then we have the internal transcribed spacer, or the ITS region, which is the official barcoding gene for fungi. And it's considered the official barcoding gene because on this website called GenBank, which is this giant database of DNA sequence data for fungi, almost every fungus, well, not every fungus, but a lot of fungi have the ITS sequence on there. So I can copy and paste the sequence from a Madagascar lichen and do a blast search on GenBank. And then it'll show me the sequence on GenBank that has the most similarity. And that will help me with a lot of things, with identifying and then maybe finding new species. Just to get into a little bit more detail about how this process works. So we take our lichen specimen and we grind it up in a mortar and pestle, and we do a DNA extraction method on that. And then we use PCR, or polymerase chain reaction, which is a very common molecular technique, which actually takes that gene, the ITS, and amplifies it, makes millions of copies of it. And then we do this metagenomic library prep, which it's a lot of using multi-channel pipettes, as you can see in that picture, and pipetting into 96 well plates. And then what we can do is take all 500 specimens and sequence them all at the same time on this next generation sequencer called the MySeq. Once we get all the data back, we use some bioinformatics to sort it out, every specimen into its own folder. And then we can start using blast searches and DNA sequence analysis to make these phylogenetic trees. And this one's like a circular phylogenetic tree that I just kind of pulled off the internet just as an example. But more realistically, they're going to probably look more, the phylogenetic trees will look more like this one here. So all 500 of these ITS sequences will be put on the GenBank. So we'll be contributing Madagascar lichen DNA to GenBank. It'll probably be the most ITS sequences from Madagascar lichens that have ever gone on at one shot. And then we'll do these phylogenetic analyses and make these trees. And in this example, this is actually a lichen from a Basidio lichen from Texas that we have found a new species for. And what we did is we sequenced the ITS gene and made a tree out of it. And the green one on the top that are in bold, these are our new species. And they actually clustered next to one that's from Japan, which is not in bold, but in the same group here, or what we'd call a clade. And I'm expecting to do stuff like that with, but I think I'm going to find a lot of new species from Madagascar using this method. And it'll also just help me identify some of these things, because a lot of these are crustose lichens, these lichens that are very hard to identify. Sometimes I don't even know what family they're in. And I'm hoping that at least with this DNA sequencing method, that I can at least get like a family or genus on some of them. And then that can help me get them to species later. There's been a lot of help on this project. Heaven Wade extracted almost all, and she's helped with this event in the past. Heaven extracted a ton of DNA for this project. And now she's moved on to do her PhD. And so now Hayden, who's up there on the top, holding a turtle, I'm trying to convert him from studying herps to be a lichenologist. And he's working in the DNA lab, finishing up with the DNA extractions, and he's actually doing this, the library prep and sequencing. And he might even be putting some on the sequencer today. And then Dylan Maddox and Izzy DeStefano, they are our lab techs that really are helping with this project a lot in the lab. And then Steve Goodman, who invited me to Madagascar, he's the Field Museum's field biologist. He lives in Madagascar. He was the one that invited me out to join in on these teaching sessions. And if it wasn't for him, I wouldn't have this project in the first place. So I'd like to thank everybody for your attention. And if you have any other questions about this project or want to see some more pictures, I have a ton of them. So feel free to write down my email address and reach out to me. And anybody that's interested in volunteering, if you're here in the Chicago area, I have hands-on stuff to volunteer with. And if you're interested in doing transcription, I could use the help, because I have a little less than, well, I have almost 9,000 specimens to transcribe right now, and we're adding more to that. So, but yeah, anyway, thank you. And I don't know if we have time for questions, but I'm happy to take them if we do.
Thank you so much, Todd. That was super fascinating. Yeah, I guess people are welcome to ask questions if they'd like. If anyone has that, you can unmute yourself, raise your hand and unmute yourself, or you can definitely type in the chat if anyone has any questions or comments for Todd. I see a question.
I am just curious about the morphology of the Bryunclaunopsis, Bryunclaunopsis, because I know multiclaula, but I don't know this guy, and I am a little curious about it.
Yeah, well, so one of my colleagues in Texas sent me the specimen, and when I opened it, it just looked like a bunch of soil, like dried up soil. And I was like, I don't know where this thing is. And then I started looking at it under the microscope behind me, and I found these tiny little basidiophores, very tiny. It's a very small one. And so one thing that's interesting about these Bryoclavulas is that this genus was first described in Japan. And so, and that was like back in 2021, so not very long ago. And so, so a research group in Japan identified or found this, described a new genus and species, this Bryoclavula phycophylla. And so he sent it to me thinking it's another multiclavula. And he's like, can you just sequence the ITS gene and see what this is? And so I sequenced it again with the help from Heaven. She was actually the one that extracted the DNA from it. And then Izzy sequenced it. And so then we blast it, and it blasts to this bryoclavula, which I had never heard of before. And so we started looking at it more carefully. And actually, my colleague in Texas was looking at it more. And he's the one that's actually writing up the new species description for this. But so like the only thing that matched to it was a species from Japan. And nothing else. It didn't blast to multiclavula, which multiclavula is in a different clade, totally unrelated, but they look very similar.
And your guy is from Madagascar or from United States?
So in that example, that's just an example from the United States. So I didn't collect any basidiolichens in Madagascar. I only collected ascomycetes. But yeah, I was just showing this tree as an example of what we could do, because I don't have any of the Madagascar data yet. I'm hoping to by next week.
Thank you so much.
But yeah, I'd be happy to share more of that with you, Luis. It's good to see you again. Luis was, he's been a visitor to the Field Museum doing research, doing DNA sequencing in the lab. And I think you're coming back here maybe, right? So yeah, that's great.
Great. And I see we have a question in the chat, Todd, from Raina saying, how long did it take to collect your specimens?
So I was in the field 10 days at each site, and we were camping in tents. And so every day we would go out and collect, usually for like 3 to 4 hours per day. And it wasn't just me doing the collecting. It was also, I was also letting the people who are learning about lichens, letting them collect things and stuff. So it's we didn't go super fast. But it was, yeah, over a 20-day period, we collected about 500 specimens.
Great. Well, any other questions for Todd before we get back to transcribing? If not, I will say again, thank you so much, Todd, for sharing your project. I will stop the recording.